future of rice functional genomics. The panel consists of Dr. Apichat Vanavijit, Director of the Rice Gene Discovery Unit of Biotech Thailand and the Rice Science Center at Kasetsart University. Dr. Edward Runge, Program Director of Monsanto Bichel Boryag International Scholars Program and a professor and Billy B. Turner Chair in Production Agronomy in the Soil and Crop Sciences Department at Texas A&M University. Dr. Chi-Fa Zhang, Professor and Dean of the College of Science, Life Science and Technology at the Huazhong Agricultural University and the Director of the National Key Laboratory of Crop Genetic Improvement. Dr. Eric Murchie, Lecturer in Crop Physiology in the Faculty of Science at the University of Nottingham, England. Dr. Ji Kang Lee, Chief Scientist at the National Key Facility for Crop Gene Resources and Genetic Improvement of the Chinese Academy of Agricultural Sciences in China. And finally, Dr. Akhilesh Tyagi, Professor at Delhi University in the Department of Molecular Biology and Director of the National Institute of Plant Genome Research in New Delhi. Our moderator will be Dr. Jan Leach, Professor in Plant Pathology at Colorado State University and a Fellow of the American Phytopathological Society, American Association of the Advancement of Science, and the American Academy of Microbiology. Please welcome our moderator and the panelists for this session.
Consolidation of the future for rice functional genomics. So over the course of the week, we've heard a lot of wonderful stories on the integration of genomic tools and technology with genetics, physiology, biochemistry, metabolomics, proteomics, on down the line. And the use of this to solve what were considered intractable problems, including improving energy capture and retention, such as C4 rice, biotic and abiotic stresses, nutritional improvements, and we've even heard a discussion on the tsunami of data that we're going to be facing and how we're going to be able to make sure that that data is accessible to a lot of people and to answer these problems and solve the problems of the future. So in this final discussion session, for a very brief time, we'd like to come back to the discussion of the first day and ask, are we on the right track to drive the next green revolution? I'd like to get some final thoughts on what is it that we need, are we prepared, and how can we best accomplish the goals that we set out to achieve. But first, one of the major problems or shortcomings that's been identified by several scientific societies and industries is the lack of training, well-trained, broadly trained students and postdocs to carry on the future of the field. And I'd like to invite Dr. Ed Runge, who's here to describe an opportunity for the Bichel Boryag program, fellowship program.
Thanks, Jan. I would like to thank IPChart for making sure that I came to your meeting, and I'm very pleased to be here. I'd like to just show you a few slides that will explain the Monsanto Bichel Boryag International Scholars Program. And basically, this is a tremendously good opportunity for students to get their PhD in rice breeding or wheat breeding. And there are two scholars here today. Mr. Ekpal is working with AppyChart, and Mr. Kim is working with Dr. An. One's from Korea and the other's from Thailand. So I'll go through some slides fairly quickly. Our mentors are Dr. Boryag and Dr. Bichel, and I was very, very pleased to be able to work with both of these people. The initial grant from Monsanto was $10 million over a 5-year period, and we're in our 5th round of scholars at the present time, and they've extended it for 3 more years. Monsanto is a large company that's involved in producing primarily maize, soybean, and cottonseed in the U.S. and other parts of the world, but they also invest in many things, and this is a tremendous program that there's just an outright gift that they've made to make sure we train the best plant breeders we can. So we need to produce more, conserve more, and improve farmers' lives, and we've been doing this in the first green revolution. Obviously, we want to do more at the present time. So wheat and rice, the reason we're involved in wheat and rice is these two crops have been increasing in production less rapidly than the population. And as a result, this is why the investment has been limited to wheat and rice. Corn is the major exception. Hugh Grant, Ted Crosby, and Norm Borlaug were involved, and I am happy to work with all these people, and I'm pleased they asked me to direct the program. I mentioned it's a $10 million program. It's a prestigious program. It's competitive, and it has to focus on a global constraint on the rice and wheat production. So what we're trying to do is to overcome the shortage of plant breeders, and we're asking them to have a dual training program. If Mr. Ekpal is working in Kasetsart in Thailand, he's going to spend a year with Julie Bailey Series in Riverside. Mr. Kim is working in Korea, and he spent a year at the University of California in Davis with a guy named Ty. So the big thing is to get the students exposed to a wide group of people and having different views on how to approach problems. So we have a judging panel. We collect the applications. This next round is due by February 1st, 2013, and the judging panel will select the group from these set of applications. So far, we've had 52 scholars funded from 21 different countries. And you'll notice that there's more from India than anywhere else, 14. The next largest is 4 from China, US, and Ethiopia. 18 are young ladies, and 34 are young men. 31 are in wheat, and 21 are in rice. So we look at different ways, but the merit of the student, the reputation of the professor, the collaborator, and these things are looked at by the judging panel to make sure that we select the best possible people from the applications that we have. And the idea to having the students exposed to 2 different research programs is to get the professors to collaborate, get the students exposed to increase their capability of knowing different approaches to the research problems and give them as good an education as we can. This is the panel of judges. Jan Leach has just been added, and she replaces Monty Jones, who had difficulty in attending our meetings. And I'm very pleased to introduce Jan to you. We have 2 that head up the wheat program, Catherine Toulet and Sanjay Rajaram. 2 head up the rice program, Jan Leach and Gurdev Kush. And then Ron Phillips, Huke Zai, myself, and Ted Crosby is an ex officio person. So I'm not going to burden you with this, but the thing that you need to write down is the website, www.monsanto.com/MBBI Scholars. You get all the information you can from there, including the application form, etc. We do not go with, the students have to be admitted to the university in which they're going to go to. We do not get involved in the admission procedures. So that gives you a brief overview of what we do, and I'd be happy to answer questions later if that's needed. Okay, thank you, Jan.
Thank you, Ed. So for the next few minutes, what I'd like to do is ask, as we did on the first day, the panelists to give very brief thoughts on our charge today. I'm going to have the bell rung at 3 minutes just so we don't ramble, as you know we like to do. And so I'd like to start working from my left to the right, if you wouldn't mind starting, Akbar.
Thank you very much. When I look for future of functional genomics of rice, I feel I could predict more accurately. but I have my own doubts about that. Still, I have a wish list of a couple of things which I would like to tell you. The first thing in my wish list, as I see, is that the present phase of data generation and analysis would possibly change into understanding and robust predictive potential, so that we are able to predict from this understanding complex questions. For example, what could be the heterosis potential of the breeding lines? So these changes are the ones which possibly would happen in the coming years. The other point, she gave 3 minutes and 3 points, I would say, relates to the marker assisted selection. And I hope that in the coming years we would see a change more from marker assisted selection to marker assisted breeding. And in this process, I would like to see that we are developing low cost and high efficiency markers. Most of the markers which we have are from genic or intergenic regions. I hope that epigenetic analysis which is going on would provide third category of the markers, and the epigenetically active regions would be used to generate certain markers which are expected to influence many traits as we feel today. The third point relates to the kind of rice. You have heard of the terms like super rice. I would like to introduce another term which we were discussing some time back, and that is designer rice. Now, what could be a designer shirt or a designer rice? And I feel this relates particularly to the nutrition and health. We see in the world that people with disease like diabetes are increasing in number. We also see that nutritional hunger is a great problem besides the normal hunger about which we normally talk. And therefore, it would be possible by breeding as well as by genetic engineering, I hope in coming years, to have rice which have appropriate kind of supply of sugars for diabetic patients, and would also be able to take care of 3 major nutritional deficiencies like beta carotene, iron, zinc, etc. Thank you very much.
Thank you. Chikang.
Okay, thank you. Yeah, first of all, I think this is a very exciting meeting. I don't have to repeat again. There's tremendous progress in the area of rice functional genomics already witnessed in the past few days. But I would like to take this opportunity to bring this group attention that in the past, most effort has been focused primarily on the mutants. And we already see the primary purpose of the Green Super Rice Project try to devote $4 million, try to sequence the panel of the core collection, is that we also, by breeding, demonstrated tremendous of the hidden diversity for complex trait exists in the exogenous germplasm, even in the primary gene pool. I think providing the sequence information, I would like to call for more effort to looking on the complex system through the new tools in genomics and other way to understand the complex system. Sooner or later, we have to deal with this problem. So, and this also brings to bridge this group to the breeders group, because we are often challenged by the other group, plant breeders. They are asking, okay, how many of the genes you cloned, you characterize has been in the farmer's field? So in that sense, I would like to say now we are providing tremendous large number of we call the integration lines as a genetic stock for everyone. Also provide the sequence data for your deep mining and free available. I hope this group will spend more time to pay more attention to this group. Secondly, I also would like to say for this conference, we probably should invite more and more people involved in breeding. so that they can understand or see what's going on in the area, so that bridge can be built by the two groups by joint effort. With that, I will thank you.
Thank you. Eric.
Thank you. Yes, I'd just like to repeat that this really is an exciting meeting to see all the possibilities that might actually come to fruition. I just wanted to talk briefly about photosynthesis, as you might predict. There's been a lot of talk about C4 photosynthesis, and if this was actually to come to fruition, we would see not just an increase in yield, but also an increase in water and nutrient use efficiencies. But it's also worth realizing that the radiation use efficiency of C3 plants still falls below the theoretical maximum, so we really should be thinking about broadening the range of targets. Perhaps in the short term we can get some quick changes as well. So, for example, one thing that's not mentioned very often is the optimization of canopy photosynthesis, and also a simple increase in photosynthetic capacity might have an effect as well. To actually do this, what do we need? Well, I think that the phenotyping is really going ahead at a rapid pace, but I think we need a sort of acceleration of the availability of phenotyping techniques, and perhaps some really new rapid phenotyping, high-resolution phenotyping techniques that perhaps can incorporate three-dimensional attributes of plants, such as three-dimensional chlorophyll fluorescence or photosynthesis, something like this, which is actually theoretically possible. And in terms of traits, I think we do need to have the ability to target combinations of traits, and this is particularly important when it comes to designing canopies, and designer plants were mentioned, designer rice, and I think that this applies to physiology and photosynthesis as well. We do need to combine essential traits, and also to continually link with models that will actually sort of embed the principles that we are looking at into reality. Okay, and lastly, I know there's been a few sort of hitches with the exchange of materials at a rapid enough pace around the world, so I think that perhaps a concerted effort to increase the ease with which material can be transferred between countries would be extremely beneficial. Thank you.
Thank you. Rod?
Okay, thank you. It's a fantastic meeting, a lot of really great science, and I want to thank AppleChart for organizing this and the entire organizing committee. So when I talked on Monday, I talked about the concept of the 9 billion people question: how are we going to supply enough food to feed the world in less than 40 years with 2 additional billion people on the planet? And we've heard many talks about green super rice, Rice 2020, which is a initiative to try to determine the function of all genes in rice. And then that led to this concept of trying to actually think really big and try to raise on the order of $9 billion to help solve the 9 billion people question. I'm very serious about that, and I think there are I've gotten many positive responses from the audience to discuss this, and I think we're going to be having this conversation openly for the next year, and hopefully we can come up with a viable plan to make this happen. And many of the talks that we heard today, or just comments a few minutes ago, they all require brains, smart people, but it also requires a tremendous amount of funding. And we only have 25 years to solve this problem, so we really need to act now. I'm on the organizing committee for the International Rice Symposium in Rice Functional Genomics, and we had a meeting yesterday, and we discussed a lot of topics. One of the topics was the possibility of actually changing our group into rather than the International Symposium on Rice Functional Genomics, would be the International Society of Rice Functional Genomics, which would actually be to form a society to help tackle the major problems that we are faced with over the next 25 years. So I think there's a questionnaire that's been handed out, and we wanted to ask you if you thought that that would be something that would be desirable for this group. One of the several advantages for actually starting a society would be that we would have a common voice for our community, for the Rice Functional Genomics community. This would also provide an opportunity for large-scale international funding. It would be a mechanism to coordinate Rice Functional Genomics activities globally. We would also use the society to run our annual meeting, and this could also be used for running and coordinating specific meetings on specific topics such as breeding, photosynthesis, and so on. And lastly, it would also be involved in managing the Rice journal that many of us have published in, and it's becoming very widely read. So with that, I'll turn it over to Apachart.
Thank you, Rod. I think Rod stole my topics, so I have to change it drastically. So at the front, so I met Rod Edwards last year. And we proposed students through the program, and we went to the World Food Prize this year. The atmosphere that looks like the program is trying to build new generation of rice breeder, a kind of to make new Norman Bollock or Bichel. For my feelings, I don't think the new generation of breeder is not going to be Norman Bollock anymore. I think we rarely find a breeder that working like the Norman Bollocks. We are seeing new generation of a breeder that learn a lot of genetics and genomics and apply for it. In this meeting, we learn a lot of topics: NGS, sequencing, SNP, a lot of things, gene function. But breeders, the real breeders, feel that we still far apart to use the work from the lab. I talk from my side that I used to be a real breeder working in the field. And I have changed myself to learn technology. So I think the new kind of breeders have to change the attitude of being the few breeders only and becoming more molecular. And then we can understand that how to use the new technology. Alternatively, scientists have to think about the application on the field side. How breeders going to use your functional gene that you discover a lot nowadays. How to make it translate into functional markers that breeder can be used, and translate the experimental result into field, the realistic environment. If we can close the gap of experimental to the field, then the breeder can enjoy our advancement. So the second point that I want to make is we want to make this meeting appealing to the public. So Bao Antonio and me try to deal with the Rice Journal. The Rice Journal have been an open access journal for quite some time and get very good impact. And now the good news is we're going to get 20 paper published free of charge. So you know the open access costs quite a lot. So these 20 papers will be something high profile, but a lot of them will get suggestion from the chair. We would like to have new generation writing good paper. We expect to have more than 20, but the first 20 will be free, and anybody who have poster session and have very good result can voluntarily submit the paper. And I will suggest that you do that by January, because we're going to publish the special issue of Rice Journal within the April, right? So please visit the Rice Journal website, and you can see the detail of this special issue. Thank you.
Future, including training the next set of breeders with new skills and new expertise. We've talked about understanding and robust predictions, moving marker selections, marker-assisted selection into marker-assisted breeding, designer rice, sequencing deeper and getting that sequence information out into the public, understanding how to get this information to the breeders and into the field, translating the information into the field. We've talked about how do we improve rice in terms of the photosynthetic rates, but also optimizing what we have to work with, the C3 photosynthesis and the capture of energy and optimization of photosynthesis and capturing light intensity. High throughput phenotyping is one of the major bottlenecks that we face, and getting these to measure the traits that we need to get out to the field. We need to capture together what are the most important traits, and a lot of that was done at this meeting. Again, we're back to the fact that the exchange of materials has been a problem for the future. And then finally, we heard again that we need to think really big, with big audacious goals about how to feed the next 9 billion people over the next 20 years. And the suggestion that we form an international society made up of the Functional Rice Genomics Group that would, in fact, take over the journal, which has volunteered to publish 20 of the papers from this meeting. So I'd like to open the discussion up to the floor and ask that you identify yourself if you come to the microphone. Are these the best ideas as we move forward into the future? And what other ideas are there? Do you have additional thoughts, or would you like to get more comment from our panel of experts? The microphones are open. You know, in classes, we teachers will point to people and ask them pointed questions. And I'm not above that.
Jan, I'd like just to add a comment. One of the areas in which we need more people from is this part of the world. Obviously, China to Indonesia, and you go east or west to India, we have a tremendous number of people, and they're based on the rice culture for the most part. So we're looking in my program, the Monsanto Beetle Borlaug International Scholars Program, we're particularly looking for rice breeding students to be nominated for support. We have no one from Indonesia, for example. We only have one from Thailand. We'd like to improve those odds and have more people, Vietnam, etc. So that's my purpose for being here, is to make sure we encourage you to nominate your students. Thank you.
Any reaction? Yeah, please.
I'm Zeba Siraj from Dhaka University in Bangladesh. So I'm just going to talk about the name, the International Society for Rice Functional Genomics. If you could add for rice improvement to encourage the breeders to come on board. So that's one. Bangladesh is not represented in this society. I don't know how much I can influence, because I'm an academician. The Bangladesh Rice Research Institute is the government mandated institute, and in Bangladesh we are not very closely knit. So I don't know how much I can influence, but I like the idea of this society, and I'll see how Bangladesh can support the society. So I'll see what I can do anyway. Thank you.
So the suggestion is International Society for Rice Improvement, which encompasses what all of us do, I would say.
Just add one comment that I expect in the future there are more and more of the effort should be devoted to translate this effort from this group to the application. So that means in the audience we have more and more probably people involved in breeding. And how we're going to do, and this is expected, if we cannot do that, it's going to be a problem. So how we take some steps to change or what needs to be done to move toward that direction.
Okay, so my name is Dabin Zhang from Shanghai Jiao Tong University in China. So I'm quite supportive for the society, so appreciate your proposal. So regarding the land, I would like to suggest more general name of the society, not specific for the rice functional genomics. It may be too narrow. If we can expand to the name such as International Society of Rice, maybe general, we can cover the fundamental research, also the applied research, including the improvement of rice. So this is a suggestion. The second point for the society is if we join the society, we do hope to share the material knowledge of rice research. So if the society can provide more help for the material or information, this will be helpful. Another point that should be considered is the difference of Arabidopsis and rice. Arabidopsis is pure model plant for fundamental research, not directly linked with applied to agriculture, but rice is different. If the mutant or gene can be used directly for breeding. So we should remember the difference, how to protect the IP in the future. Thank you.
Thank you. Go ahead.
Okay, Antonio, University of Pelotas, Brazil. I would like to congratulate all the speakers. I think they're very important points. And recently in August we had a meeting of the International Crop Science Society, and it was also mentioned the lack of breeders that we are, or the training, we need more training for breeders in several crops, in this case here today, for rice breeders. Maybe we could add perhaps in future meetings a session for this Borrow Fellows to come and present their progressing work, so we can see how our functional genomics is translating into breeding on a specific initiative, or that would be an idea.
Excellent. Any thoughts from here?
I like what Dabin said. I think maybe and like broaden, make it bigger. Rice science or rice research, make it big. And my question would be, how many societies for rice exist today? And maybe this international society could have in each country a society that can mirror it to facilitate exchange between breeding, genetics, and functional genomics. But and I have no idea if there are any societies in any country for rice research. So that's a question to you.
I honestly don't know of a society specifically for rice. I know there's the Rice Genetics Congress, but we really haven't. I'm not sure of a society for rice, but that's kind of why we brought it up. I mean, it's a lot of work to actually organize a society, and a lot of people were backing off. But I think the thing is this is so important and we have to act. And if by having a society, then we can actually do some of these big things that we're talking about. So I think a lot of people I've talked to are in agreement with that, and it does make sense to maybe have national societies that move into the international society. Thank you.
I think there's a foundation called the Rice Foundation, and we have the Bangladesh chapter of the Rice Foundation. I know that, but I'm not sure who runs the International Rice Foundation. I mean, it's just called Rice Foundation.
Thanks.
Familiar with it, but I'm not sure how it's managed or run.
I know there's one problem. Next year will be a rice genetic symposium. Then there will be a rice functional genome symposium. It will be separate. If that's the case, because the rice genetic symposium, you have breeders and also a lot of other group. Now, if we separate this group with the others, it's probably not a good idea. It's not, because if you attend one meeting, these days too many meetings. I guess this group or the committee should consider how we can do to think about it to be more efficient.
Historically, there has been a link between the rice genetics, at least alternate years. Is that correct?
Yeah, that's every 4 years.
Every
now,
4 years.
But next year will be overlap. But I heard you were separate.
Anybody have any ideas on how to tie the rice genetics group with the functional genomics, with the breeders, with the rice community as a whole, and to attract the rice community as a whole into the fold? Dr. Lung.
Just wanted to explain why the functional genomics meeting separate from genetic meeting for next year. For the past 10 years, there have been 2 events that is a genetic meeting is traditionally organized by IRI and always based in Sydney. And we did it, the 2 meetings together. In number 3, I think is was the previous one. So twice we did that. And some comments coming back is that maybe the meeting is too big, too many people. I think the last time we had was like 850. So that sort of led to the last year's discussion in Taipei that probably 2 reasons. One is several countries really wanting to sponsor the functional genomics meeting. That's one. And that's been the case for the last 10 years, that a lot of countries line up to support this symposium. And then the other one perhaps is we should try to uncouple and see how it goes as a one time, and then maybe when we realize that maybe it's better to come back together, then we still have opportunity to come back together. But if we don't do it once, we always have this constant, not tension, but see, because every time if we do the genetic meeting, always in Philippines, it's not like rotating around the world. Whereas the functional genome meeting, been crossing different continents and making sure that Europe will have a chance to do it, and India and so on. So that's the reason why we separate. There are clearly pros and cons in separating two meetings.
Add some comment. I think what Hay said was a very good reflection of what we discussed in the meeting. So I think what I would like to propose further for these 2 meetings to meet from time to time, not every year, every few years. I would suggest we would consider very seriously the possibility of rotate that rice genetic meeting in a different place, moving somewhat outside from time to time, just like this meeting, a few years, one meeting several years in a different continent, different countries, so whomever interested in hosting the meeting. So, hey, I hope you could you don't need to respond to me now, but I hope that is some sort of possibility during the campaign.
I think, Stephen, that's actually a good idea. In fact, after the last meeting in Manila, Kim Doberman, our director for research, actually saying that our facilities, conference facility in Manila, is not necessarily enough to hold, say, 800 people. So there was discussion since then that we may hold it in Singapore, for instance, having a convention facility. So, Chifa, that is not something that we didn't think about. Now, this year, I don't know. I mean, I was not that actively involved in organizing that, but definitely the idea that genetics meeting, does it have to be always in the Philippines? One reason we've been doing that is from the standpoint of ISRI. There are many young scientists wanting to see, having seen ISRI as an institution and going to the field and then seeing how ISRI conducts its business. That is a plus. So that's, as I said, there are pros and cons about doing the meetings in Philippines. But doing rotating genetics meeting in other places in conjunction with functional genomics is not a bad idea. That's just my personal opinion. Whether when I go back home, whether people like the idea of breaking this tradition of having genetics meeting in Philippines, I don't know. I'm just voicing my personal opinion.
Well, I think that the idea is to get ideas on the table that we can do as a community, and inviting the geneticists and the breeders and the functional genomics people to meet together is one idea that we can put out as a group. Other ideas, other suggestions on how we can move forward as a community, whether it be as a society or as a community to solve the problems of the future, or their shared approaches. So I think we've had several good suggestions. We've had suggestions for how to approach becoming a more consolidated community, one of which is to form an international society, and there's a plea that this be a broadly representative society, not just genomics, that it reach out further, and that we embrace the translational components of our field. Are there other suggestions or comments from the panel? No. Questions you'd like to raise? Ask the community while you have them here. Any other comments from the community? I know it's been a long week and you've had a great time. Well, if there are no further comments or topics that are to be brought up, I'd like to take this last opportunity to thank Apishar and the whole team of folks that have manned these meetings, and the students, postdocs, staff that have helped him to pull off what I consider to be a really excellent meeting, and I think all of you would agree with that. So if we could just have a thank you for you.
Thank you very much, Dr. Leach. Can we have a round of applause for our panelists and our moderator for this session? Now, please just give us a moment while we clear the stage in preparation for the closing ceremony. Time for the closing ceremony for the 10th International Symposium on Rice Functional Genomics. It is my pleasure to invite our organizer, Associate Professor Dr. Apichat Vanavitit, to express sincere thanks and appreciation to all of you and our distinguished guests. Please welcome
many
Dr.
expertise in order to explore our knowledge and exchange ideas and strategy leading to latest information and technologies according to our theme, driving the next green revolution. At this time, I am most grateful to give my deep appreciation for all distinguished guests participating in this symposium. Plus, I hope we will meet again next year. Now, it is my great honor to present the overview of our next host for the 11th International Symposium of Rice Functional Genomics in 2013. Please welcome Professor Tayagi to the stage.
I hope you all will agree that we had a gala time here, both academically as well as otherwise. And it is the time that we thank EpiChart and move to the next stage. In this situation, I stand in front of you on behalf of rice researchers community in India and would like to extend invitation on behalf of Indian rice research community as well as my colleagues from National Institute of Plant Genome Research and Delhi University to 11th ISRFG in 2013, and we plan to hold it in New Delhi towards the end of November. Exact dates for the same would be communicated to you very soon. As you all know, India is a rich center of diversity in rice, and our scientists are busy in classical breeding programs and also in introgression of new traits by using molecular tools, as exemplified by the golden rice which is being introgressed into rice varieties in India. Thus, in addition to science, you would also have an opportunity to visit New Delhi, which is the capital and the third largest city of India, and it is a fusion of ancient and modern Indian culture. The city is very rich in very old monuments and has a culture of more than 3,005 years. Sometimes it goes to prehistoric times, and you would be able to enjoy a combination of both of these. Also, it is academically very active. There are about 20 central, state and private universities, and there are another 100 academic institutions within New Delhi and what we call as National Capital Region. I am sure that you would be having some time, and we were discussing there would be some time to visit monuments like Red Fort, Jama Masjid, or Digambar Jain Temple, symbols of the unity of the city and the country which we call India. We also have the New Delhi, which is also a seat of government's activity, and I am sure you would enjoy visiting India Gate and the adjoining areas, including the Parliament House. Of course, we cannot forget our culture of dance and the variety of the dances which are available. I hope there is a possibility to watch some of them. And most important, and a symbol of India, the Taj Mahal, if you spare a day, you would be able to visit Taj and return in the evening. With this all, once again, I would like to invite you and your colleagues who are not here. Please communicate to them. We extend our invitation to all of them to visit India next year for ISRFG 11 in New Delhi. Thank you very much. There are some handouts, and also please note the email address through which you can contact us, and that is isrfg11@nipgr.ac.in. We would also provide you information through our webpage www.nipgr.ac.in till we have a dedicated site created for all the activities of ISRFG 11. Thank you very much.
Thank you very much, Dr. Tyagi, for that inspiring presentation. And with that, the 10th symposium now draws to a close. On behalf of the present organizer, I would like to thank all chairpersons for hosting the sessions, all speakers for your presentations, and everyone for making this symposium a vibrant forum for mutual learning as we move forward together. All presentations will be made available on YouTube via our website. Please inform us if you do not wish your presentation made published. Also, our website will be active and you can find galleries and abstracts there. I would also like to express our appreciation to our sponsors for their contributions. This event has been generously sponsored by various companies, namely PCL Holding, Bio-Rad Laboratories, Primus Scientific, Gene Plus, Eppendorf Thailand, RI Technologies, Biogenomic, Lab Focus, Bioactive, Delta, Gibtai, and LECO. Thank you all very much, and I hope that our hospitality has not been lacking during your stay here. We take great pride in presenting to you now in farewell 2 dances in the Northern Thai tradition, and hope that they will leave you with warm impressions of Chiang Mai as you depart for home. Farewell, and see you all again next year at the 11th International Symposium on Rice Functional Genomics in New Delhi. Thank you.
电费。